All right. Well, thanks, folks, for coming. We'll get underway. It's a really great pleasure to introduce Jeff Hanson to you. Those of you who've been around a few years will remember Jeff, of course. Jeff did his PhD here a number of years ago now. When did you finish? 2020 or something? No, before that. He doesn't even remember. So much has happened since then. Jeff did an absolutely outstanding PhD here, focused around, some of the work he's going to talk about it today, I think, focused around prioritisation. He published 4 papers, all 4 of his PhD chapters were published, and Methods in Ecology and Evolution, Journal of Applied Ecology, PNAS, and Nature, which gives you an indication of the sort of quality of work Jeff does, and the depth of his thinking and innovation in the whole field of conservation prioritisation. Jeff has really made a number of really important scientific steps forward, but he's also built a really important piece of software called Prioritiser, which enables one, with others, but Jeff has been a leading light in the formation of Prioritiser, which enables one to operationalise many of the things that Jeff's going to talk about today. So, you know, you've got a chance to actually put some of what Jeff is talking about here into action yourself with the brilliant Prioritiser package. Since his PhD here, Jeff did a postdoc in Portugal with Silvia Carvalho, working on genetic variation and prioritisation along environmental gradients, and now he's doing his PhD, a second postdoc at the University of Carleton, as he described earlier, and I'm sure we'll talk about some of that work too. So Jeff, it's a great pleasure to have you back here again. Thanks for coming to visit, and we're really looking forward to hearing your talk. Thanks so much.
Thank you so much. Yeah, it's great to be back and see everyone. It's really cool. So yeah, I'm going to talk to you today about conservation decision making, and maybe you'll find it interesting. So before I get into it, I'd first like to thank all the people that have contributed and played an amazing role in collaborating in some of the work I'm going to talk about today. And also the various institutions that have been involved. So massive thanks to everyone there. Something in this room. oh dear, no, that's yeah. So this thing is getting old, so we're just going to jump forward, and I'm just going to have to deal with this.
Let's jump together and find a new
Okay. So we're in the middle of a biodiversity crisis. Over the last few centuries, a great many species have gone extinct, and we're at this point here where we can either do something, and if we don't, you know, things are going to get worse. And in conservation, we ideally want to halt and reverse these declines so we can get up here, so we can improve things. And the question is, how do we achieve that? So I'm going to talk about how we can make better decisions about conserving biodiversity, and I'm going to talk about 3 potential ways you can do this. There's many other ones, but this is what I've been working on. Okay, so what do we mean by conservation decision making? So as conservation scientists, we like to believe that data should play some role in how we conserve biodiversity. So we might have some observations of where we've seen different species. We can take this data and turn that into information. So species distribution maps that are showing us where we think different species occur, or different ecoregion types. We can then use this information to create a plan. So if we've got a study area, we've gridded it up into potential places we can put protected areas. These grey squares here could be places that we might want to establish protected areas based on this information. Critically, when we're making our plan, we should account for our objectives. So what we're trying to achieve, we're trying to conserve threatened species, improve ecosystem coverage, ecosystem services, and we also have to think about constraints. So that might be things like where we can and can't put new protected areas based on existing land use. Or it could be things like financial constraints, because often we have limited resources when trying to conserve biodiversity. And ideally, what happens is you have a plan, and given a lot of assumptions, we might get it implemented, and that achieves an outcome which changes the trajectory of what we think of biodiversity. So how can we improve this process? No. This is really clearly. So, yeah, that might be better. Okay, so one option is we can use better algorithms. So if we use better algorithms, this will change our plan, and that potentially gives us a better outcome using the same information. So how do we go about deciding where priority areas for conservation are? So if you've not done spatial prioritization before, I'm just going to give you a quick intro. Let's say you had a study area you were in charge of managing, and you had 8 species that you cared about. So represented by these different circles. We'll call these features. That's just jargon for stuff we care about, essentially. And we can grid up this study area, and each of these grid cells represents a place that we could select or not to establish a new protected area or implement a specific conservation action. And the question is then, which of these grid cells should we select for our conservation action? So in order to frame this as a mathematical problem, we need to think about our objectives and our constraints. So we might say that we've talked to some experts and they tell us that each species needs to have at least 1 grid cell protected, and that might be enough to guarantee their persistence for another 50 years. So based on that knowledge, we know we need 1 grid cell per species. Often in conservation planning, we have this idea that we want to minimize opportunity costs or minimize expenditure. So in other words, we want to have cost-efficient solutions. So given that, we'd want to minimize the total number of grid cells we select, assuming everything costs the same. So now we've got a well-defined problem. We want 1 grid cell per species, and we want to minimize our number of grid cells. What is the optimal solution? If you guess this, then you would be correct. Of course, it gets much, much trickier when you're dealing with hundreds of thousands of grid cells or planning units, and when you're dealing with thousands or tens of thousands of different features that could represent different species or ecosystem types or genetic stuff. So to help us identify really good solutions, people have been developing a lot of different tools and using a lot of different types of algorithms to find good solutions. So if you've heard of the zonation decision support tool, that uses a heuristic type algorithm. Broadly speaking, the general way that works is we start somewhere and then we try changing the solution and altering it. And the idea is we want to keep on altering it until we hit a peak where we can't alter it anymore easily without degrading the quality of the solution. And that's really good because that's very fast to run. But the issue is we might miss out on peaks later on because we didn't start in the right place. If you've used Mark-SAN, you might be familiar with simulated annealing. That's a type of algorithm called a meta-heuristic algorithm. And that's really cool. And so the idea there is you start somewhere and then you alter it. But unlike heuristic algorithms, you're sometimes willing to accept moves that degrade the quality of the solution. And that's really powerful because that lets you, explore these valleys so you can get to a higher peak, basically, broadly speaking. For a long time, these have been the main kinds of algorithms that people use in spatial prioritization. But there's another type of class of algorithms called exact algorithms. And these have a dual component to them. So one component tries to improve the quality of the solutions, and another component tries to guess what is the value of the highest peak. And when these two components converge, then you know that we've found the optimal solution or an optimal solution. So that's really cool because then you know exactly when you found an optimal solution, when you found the highest peak, or when you're close enough to the highest peak that it's good enough. So you may not need to be right on the top here, but you might be willing to say, within 5 or 10% is good enough. And having that guarantee is really powerful. And there's a lot of different tools that are available, such as Adobe, CFlex, or CBC, or Symphony, that let you use these algorithms. So me and a bunch of other people, we thought exact algorithms sounded like they could be useful. So we wanted to try and develop a tool that would help people use them more readily. So we developed an R package called the Prioritizer R package. And the general idea behind it was that you should be able to tell it your data, so your biodiversity data, your land use or economic data. You should be able to feed in your objectives, so what you really care about. So when you're trying to, what's the main metric by which you compare things, and then also have constraints. So that's in telling things what the solution must or must not do. So the solution must have a certain amount of habitat for a certain species, or the solution must have certain places selected like existing areas. And so the idea is you feed all of that data and problem specification into the tool, and you have a mathematical optimization problem that's defined. Then that's sent to a solver. So then we take that and we turn that into something this software can understand. And then Prioritizer takes the raw outputs from this and then turns that something into you can understand, like maps and calculate statistics and stuff like that. And so that's on CRAN, if you want to check it out. So Richard Schuster, a colleague of mine, he ran some analyses to compare the performance of various exact algorithm solvers compared to Mark-SAN. And what we found was that the exact algorithm solvers can get you better performance. So on the y-axis here, we've got deviation from optimality. So the higher you are, the worse the solution is. So the blue and the red lines, those are two exact algorithm solvers for Rovi and Symphony. And the green line is Marxan. So we can see, you know, Marxan is really good because it's a fantastic tool and you can use it to get solutions really quickly. But if you really want high quality solutions, then you might find that you might need to use a different software, which is, you know, there's nothing wrong with having lots of tools available. So that's kind of cool. We also tried recording the runtime. So we also recorded the runtime of these solvers under a range of different settings. You can think of the x-axis as settings if you know what a target is. And we found that under different settings, if you do a full proper analysis, you can actually find that using Marxan can be slower. But you know, one caveat here is, you know, some of these softwares, they cost a lot of money to use. So if you're an NGO and you need to use tools that are freely available, then you know, Prioritizer may not be for you, and you might find better performance with Marxan. But if you can access these more expensive tools, then Prioritizer might be a useful alternative, if that makes sense. So pros and cons is what I'm trying to say. So what about other conservation problems? So we've talked about reserve selection. So that's, you know, picking priority areas for conservation or protected area establishment. But we have lots of types of problems in conservation, which is really exciting because it means there's lots of stuff to do. So one example is prioritization of recovery projects. So let's say you're a manager, you're in charge of managing an area. Across this area, you might have populations of penguins, elephants, and goats. So, you know, if you want to manage the penguins, you might need to do some, kill some rats with some poison, and that's good for the penguins. If you want to conserve your elephants, you might have to establish some fences. If you want to conserve your goats, you might have to do get rid of the rats and also do some habitat restoration. So each recovery project has a defined set of actions that need to be implemented in order for that project to succeed. And of course, we always have the option of doing nothing as well. So that's important to consider. So each recovery project has a probability of being successful if it's funded. So just because we do an action doesn't mean we get the outcome we want. For instance, we could plant a bunch of trees, and if there's an extreme drought, they could all die. So we fail to achieve our habitat restoration. Assuming that we are successful in these recovery projects succeeding, we would expect to see this improve the probability that our species would persist. So if we fund the penguin recovery project and it's successful, we would have a 95% chance of the penguin persisting into the future. So that would often be guessed using external elicitation, that kind of stuff. But if this wasn't funded, then we'd have a 40% chance of penguins persisting. So we can see different recovery projects give us different probabilities of different things persisting. And generally that improves over the do nothing scenario. Okay. So in addition to our species, we also have to think about costs. So what are the costs of the different actions? And then finally, what our budget is. So what I'm trying to say here is prioritizing recovery projects is a tricky thing. And we were wondering, you know, could exact algorithms give us useful benefits here as well? So if you had this data, then that would be the optimal solution, where you fund the penguin and the goat, and you do nothing for the elephant because you don't have enough resources to implement all three of them. But you know, that's assuming you value each species equally, right? So if you cared most about the elephant because that provided certain tourism ecosystem services, then you know you go all in on the elephant, for instance. So getting back to objectives, you need to measure what you care about, and that can change what you end up doing. So in order to see whether these algorithms could do well for project prioritization, we got a hold of an amazing data set that had previously been compiled for recovery projects in New Zealand. We got a hold of recovery projects for 62 imperiled bird species. So this is a phylogram showing all the birds in New Zealand. And the yellow ones are the ones we're looking at today. So that's what 62 are. So you can see we've got a good spread of the whole evolutionary history of bird species. And for these 62 bird species, there was over 1,000 different individual actions associated with their recovery project. So that's a really tricky problem to explore. And you know, many of these actions were shared between different species. And so we looked at 4 different algorithms. So, okay, this is tricky, so I'm just going to walk you through it. So what we did was we generated a series of different prioritizations under different budgets. So if you can imagine a series of dots along here, and we've got these lines connecting those dots. So we generated a series of prioritizations using exact algorithms, and that's what this white line is showing. Yeah. So if we're higher on the y-axis, we've got a better outcome. So if we generate a prioritization using 20 million, we get a better outcome than if we have 10 million as our budget. So we also looked at ranking and heuristic algorithms. So ranking algorithms are very common because they're easy to communicate and also very easy to run. So basically, the idea is you have some measure that you calculate for each recovery project, maybe some measure of cost benefit, and then you rank them, and then you basically just fund them down the list until you run out of money, or until your budget runs out. And that can be useful in some situations. But we can see here, we look at the orange line, and also and the heuristic algorithm, that's our hill climbing one we talked about before. So that can be useful as well, but it doesn't necessarily always perform best because it doesn't, it doesn't find those peaks. So what we can see is under some budgets, our white line perfectly overlaps with our blue and our orange line. What that means is there are certain cases where these other algorithms are giving us optimal solutions, but there are also cases where they're not. So you can see this red line coming out here. And what's happening here is basically the algorithm's got stuck and the next project in the queue is really expensive. So because the next one in the queue is really expensive, it has to wait until our budget gets there. And that's why it just sits there for a while. And that's because these algorithms they add stuff in, but they don't swap stuff out. So, what I'm trying to say is if you use these heuristic and ranking methods, you can end up with a solution that's not very good. And the blue stuff, that's randomly funding projects. So this wavy blue line here is the median, I think, the median performance of randomly funding projects. And you can see that's quite low. And that makes sense. If we're just randomly funding things without thinking about anything at all, we're probably not going to do so well. But what's really interesting is we can see the blue silhouette is the range, the minimum and the maximum. So sometimes we fund projects that are really bad and basically give us nearly nothing in terms of our benefit, but sometimes they actually perform better than our heuristic and ranking methods. So failing all else, you could probably just generate 10,000 random solutions. If you didn't want to bother using the complex optimization algorithms, that might give you something that's actually better than the ranking or heuristic, which was interesting to me at least. And so if you want to try this, that's on the OPPA package, and that's also on CRAN. Okay, so that's better data. We also have, we talked about algorithms. So, data gives us information, gives us a plan, gives us an outcome. If we have better data, then that can potentially, better data, such as genetic data, that can help give us a better understanding of biodiversity. We can use that better understanding to improve our plan. So instead of using the gray stuff, now we might choose the green stuff instead with this updated information. And that gives us a better outcome, ideally. And so what did that look like? So in my previous postdoc in Portugal, we got my colleagues collected an amazing amount of genetic data across. So this is Portugal and Spain, if you're bad at geography like me. And so the red stuff here shows the distribution of these species, and the black points are where they collected genetic data. So they went out and collected this data across the entire distribution of each species, and we could use that to characterize spatial patterns of genetic diversity really well. And then we could use that to see what effects collecting genetic data has on our conservation planning process. So I'm going to show you what we did for one species, but we did this for all 3 species. So we tried to characterize metrics related to adaptive processes as well as neutral processes. So, we tried to identify adaptive clusters. So these spatial areas that contain individuals with similar genetic patterns that might be reflective of adaptive genetic variation, so like adaptations, but that's with like a huge asterisk. If you want to know more about that, you can talk to me later. But we also looked at climate refugia. So these are places in yellow are more likely to stay stable in the future under climate change. So if we want to promote adaptive processes, then that might be good to secure these areas. If we wanted to, we also measured individual heterozygosity, which is a very coarse measure of genetic fitness, again with a huge asterisk and a bunch of assumptions. So you can talk to me later if you want to know what those are. But broadly speaking, we want places with higher individual heterozygosity. And we also match patterns of neutral clusters. And I should have said this right at the beginning, but what we did was we also ran an analysis at the very beginning where we took the species and we subdivided them into independent evolutionary units or genetic lineages. So, there's a north bunch of populations and a south bunch of populations of the species. And those are kind of independent. So if you want to conserve the evolutionary history of the species, you need to get both. So what we did was we generated one plan using the distribution data I showed earlier, as well as all this genetic stuff that we calculated. And then we generated another plan without the genetic and evolutionary stuff. And what we found is they're kind of similar, but if you use the evolutionary stuff, you get some key differences. So there's certain places in the study region, so where I've highlighted in these yellow circles, where there's far fewer planning units selected. And as a consequence, this prioritization over here that wasn't generated using that evolutionary data is missing those places, and as a consequence, it's not meeting targets, or it's not safeguarding, the individual heterozygosity stuff. It's not getting climate refugia, and it's not getting the adaptive neutral clusters. And a common concern is when you try and throw in all this extra criteria into spatial prioritization, it's going to massively increase the cost. But what we found was this plan cost 9% less than this one when we measure cost as the proxy based on anthropogenic impact across selected areas. So what I'm trying to say is, if you use all this extra information to refine your conservation plan, it's not going to necessarily massively increase the total amount of area or total cost associated with implementing that plan, which is good to know. But more data isn't always better. So, getting back to this idea of data, information, plan, outcome. In theory, if we had a set of surveys and then we conducted more surveys, so let's say we went here and we saw, we tried to see what was here, that would help fill in the gaps in our distribution maps. And that could potentially radically alter our plan. So maybe these places down here are really cheap and they also have a lot of biodiversity we just didn't know. So if we knew that there was those species there, then that, we might actually select a lot of planting units there, and that could actually increase our outcome. So, another way is more data, better outcome. However, collecting data isn't free. And if we went overboard and we collected, we sent out field surveys to sample all of the missing regions, we'd have a much better understanding of where the species are. But now we'd have much less resources to implement our plan. So if we had a budget of $100 million and we spent $99 million on collecting data, we've only got $1 million left over to actually do conservation. And if we care about outcomes and not having better data, then that's not so good. So we can actually have a worse outcome than if we just, just started with what we had. But that's not to say that collecting data can't improve your outcomes as well. So to try and explore this idea, we got a hold of a data set in Middlesex County in Canada. So this is work from my current postdoc. So we got data for 90 peril plant species. So this is high quality data collected by botanists. They went out to a whole bunch of places and reported is it present or is it not present. We identified 142 places in the county that could potentially be acquired for protected area establishment, and we got land acquisition costs. And of those 142 places, we identified 90 that could potentially be surveyed to find whether these 9 species are there or not. And so what's really cool is we've got land acquisition costs, but we also have costs of surveys as well. So, we've actually got estimates of all the different costs that we're considering here when we're thinking about value of information. So the idea of going out and collecting data has been around for a long time, and lots of different people have come up with different strategies for actually doing this. So I'm going to talk about 5 different methods. I know the plots are really tiny in the back and I wouldn't be able to see them, so I'm just going to bear with me. So down here, so each column is a different approach for generating your sampling design plan. And each row is when we've selected a different number of grid cells based on a budget for collecting data. So down here, we've only got 1 red grid cell, and I'm sure no one else can see where that red grid cell is, but just believe me when I say that there's one red grid cell in each plot down here. And up here, we've selected 90, so 90 out of the 92, I think, grid cells for sampling. And in between, we've got a gradient. So here we're selecting 31 and 61. So we actually generated the full suite. So we went from 0 to 90, and I'm just showing the sample here. So we've got geographic based sampling. That's where you spread your survey effort out equally across the study area. There's environmental based sampling, where you try and sample different environmental types based on the idea that different species occur in different environmental types. So if you cover the different environmental types, you get all the species. So that's some fun biogeography stuff. This approach here is based on uncertainty. If you have fixed species distribution models for your species, you can actually quantify where the model is most unsure about whether the species is there. So finding a place where the predicted probability is 0.5. If the predicted probability is 1, the model's really sure that it's there. If the probability that the model goes to 0, then the model's really sure that it's not there. But when it's close to 0.5, that's when the model really doesn't know. So you can find those places that are 0.5 for most of your species, and that's the place that the models you're most unsure about. Another approach is really interesting. So this involves just sampling the places that have the cheapest land acquisition costs, right? If you've got here we have an extraordinary range of really expensive places and really cheap places. If we find one of the imperiled plant species in one of the really cheap planting units, then it's a no-brainer that we'd just protect that because it's an easy win-win. And finally, there's the richness-based approach, which is sampling places with the highest richness of the species you're interested in. So the idea there is, you're double checking the places where your model predicts that most species are likely to be. So you're double checking to see whether the species are there before you actually do your management action. So you're double checking your information. We also did some optimization stuff, but let's go through how we can use our value of information framework that we developed. We can use that to measure the performance of approaches. So we'll do that first. So when we did these analyses, we looked at 4 different budgets. So assuming a total budget of 7 million, 12 million, 19 million, and 26 million. So on the $7 million budget, that $7 million are combined for collecting data as well as acquiring land for conservation, and the same for the 26. So we can calculate something called the expected value of current information. So this is how good our conservation plan would be if we only used existing data. And that's represented by these gray dashed lines. So further up on the y-axis, the better our conservation outcome, so the more species we're adequately representing. So, if we've only got 7 million, we expect to get about 2.5 species adequately conserved. If we have 26 million, it's about 6. So basically, if we have more money, we get a better conservation outcome. That's not really very insightful, but that basically shows that the math kind of adds up a bit. So let's look at one of these approaches, so the geographic approach. So here what we can see is, so we generate a series of sampling designs across, so you can imagine lots of dots along here and they're connected by the line. So down here we've got for a sampling design where we select one site, and here we've got a sampling design where we select all sites. And we can see under a budget of $7 million, it kind of stays here and then just decreases. And the reason for that is here we're spending $7 million on our surveys, and our total budget is just over $7 million. So here we're spending the vast majority of our funding on collecting new data, as opposed to leaving very little left over for implementing conservation actions. So naturally we get a worse outcome. Here we've got a much higher total budget. So the same strategy of sampling everything can actually be much better. And that's because we've still got plenty of leftover resources to actually implement, to actually use the information to implement conservation actions. So what this says is the performance of a sampling design really depends on how much resources you have to begin with, because that'll reflect how much money is left over to do things. So now let's look at all the other approaches we looked at. So we can see straight off that under the total budget here, we've got a bit of variation here, and then basically everything, no matter what strategy you do, if you're spending lots of the most of your funding on collecting data, that's not going to give you a very good outcome, because you're not going to have much money left over to achieve, implement conservation actions. And we can see a similar kind of pattern here where we get a bit of variation, and then eventually they'll converge at the end. So what was interesting here, this red line, that's our management cost approach. So that's where you're just surveying places with the cheapest land acquisition costs, which is really surprising because often we'd like to think things based on biogeographic theory might be useful for conservation. But it turns out this simple rule of thumb worked really well in this study system, which is possibly due to the fact that there's such a large range of protection costs. Maybe if you've got a study area where you've got more homogeneous land acquisition costs, then maybe you might find that these other approaches like geographic coverage would be better. But we can see critically, no matter which approach you use, if you spend too much funding on collecting data, you're still going to get a worse outcome. So we also embedded all of this within an optimization routine. So we used the heuristic algorithm we talked about earlier. I spent a long time trying to get this with exact algorithms, and I couldn't do it. So if you're a maths genius, please talk to me, and you can tell me how to do it. But yeah, so what we found is, so we generated an optimized sampling design. This one cost $1.57 million. So I've shown it with a dashed line, but you can probably think about it as a point that occurs about here. So what we find is with this optimized routine, it's not, this is the best we could do using this algorithm, and it improves our current information. And what's really useful here is we know that we don't have to explore a range of different budgets. We can just run it once, and that gives us the best, I should say the best, a near optimal answer. And we can see that the cost of the optimized survey scheme, that changes depending on our total budget. So, that's kind of cool. What we can also see is the gap here changes depending on our total budget as well. So, it's a bit smaller here, whereas it's a bit bigger down here. So what this suggests is the potential room for improvement that you have for conducting your, for collecting new data might depend on how restricted your resources are to begin with. Phrased differently, if you've got very little budget, then maybe the best thing to do is just spend all that on achieving your outcome rather than collecting lots of data. And of course, your optimized method does best because it actually considers, it's measuring this and trying to improve that as much as possible on the y-axis. It's not using some kind of method to get around that. So that's really cool. We can go out and collect data and that can give us a better outcome potentially. But often we don't have the luxury, or if you don't like going outside at all and you'd rather just download data instead. So, so we also have this idea that you can use surrogate data sets instead. So the idea with this is, you might have a gold standard data set that you'd really like to get, like field data, but maybe you can't get that. So is there other data sets you could use instead as a surrogate or a proxy to try and get the same answer or the same kind of information if you can't use that gold standard? So I'm going to walk you through some basic evolutionary stuff if you're not too familiar with that, because I'm going to talk about surrogates for evolutionary stuff. So let's say you had a species shown in red, so it's like the range. You've got a set of populations. Let's say these populations were living across environmental gradients. We've got forest type habitat here, desert type habitat over here. In time, and given a lot of assumptions, we might find that the populations in the forest type habitat develop forest type adaptations, and populations in the desert type habitat develop desert type adaptations. Again, lots of assumptions. And we also might find if there's a dispersal barrier like a mountain range, then that'll cause neutral genetic processes that will mean that these populations become differentiated from the other ones. So if we wanted to conserve the full spectrum of within species variation, we want to be getting the different types of local adaptations, but also getting different evolutionary units too. And so ideally we'd have genetic data to do that, but we don't always. And so of course, if we have this genetic data, we can use that to create our plan, give us an outcome. But if we don't have genetic data, then maybe we could use environmental data. So this is one of the ideas that we looked at in my PhD. So if you don't have genetic data and you have reason to believe that local adaptation might be happening, you might be able to use environmental data, which is readily and freely available all over the world instead, and that could be really useful. So the idea is, instead of using the genetic information, you use the environmental information instead to create your better plan. But that hinges on this idea that we've got a good enough understanding. This gives us a good enough understanding, and if that assumption is met, then all of this is potentially wrong. So we need to verify that. So can we use environmental and geographic variables as surrogates? So we, some previous people had gone out and collected an amazing data set across the European Alps. So, this is the European Alps here. Down here is like Italy, I think. And so what we have is this amazing data set of 27 plant species with different life history strategies, different niches, and different dispersal stuff. So it's an amazing data set. And, the authors who originally collected this data set did a fantastic job. And so what we did was for each species, we mapped patterns of potential adaptive genetic variation. So what we found is, so for each of these grid cells, they went out and collected samples, and then so we can actually characterize what's happening within each grid cell. And so we can use that to say, well, for this species, there might be a north-south kind of gradient in adaptive genetic variation. And we use some analysis to look at different gradients of that variation. So that's why there's multiple panels. So that's the dominant one for this species. For this species, it had 3 main axes of neutral genetic variation. So you can see like an east-west kind of transition. And for these other ones, they can go south and some other stuff. And in case you're interested, this is what the environmental variation looks like across the study area. So we've got a north-south kind of east-west, and then something else is going on here. But yeah, so that's kind of what that looks like. And so what we did was, so we did that for all 27 species, I think. And then we generated 3 prioritizations. So three conservation plans. One was just using the distribution information, so only using information of which species are in which grid cells. One using that information plus our surrogate variables, so using environmental and geographic variables as a surrogate for the genetic variation. And then also another prioritization that was based on using the distribution data as well as the actual underlying genetic data. So what we can see immediately is, if we don't use any surrogates or we don't use any genetic data, this plan is much fewer grid cells selected. And so that says if we don't account for within species variation, we might underestimate the scope of conservation actions required to conserve species. But what we're really interested in, is how well do these perform, not just the spatial arrangement, because you can have different spatial arrangements that give similar performance in terms of how well they achieve objectives. So what we did was, for each of these 3 prioritizations, we measured the amount of genetic variation they secured for each of the 27 species. These are box plots, but because they're all really close together, it looks like a line. Yeah. So we can see here that this prioritization did really well at getting adaptive genetic variation for most species. This red dashed line is our benchmark, it's our target. So if you're over this, you're doing really well. So we can see if we use just the distribution data, we're doing well. If we're using the distribution data plus our surrogate variables, you're also doing really well for most species. This species down here that was kind of just below the red line, that's now above. So it's still an improvement. And we can see here if the genetic data reports are above the red line. So this is really exciting because it means you may not necessarily need to account for even surrogate variables in order to get adaptive genetic variation. The caveat here is though, our system, we were looking at 27 plant species with such a broad range of different habitat specialists, habitat generalists, and all that kind of stuff. So that means that prioritization was forced to represent effectively different environmental types because we have so many different environmental specialists. And that means we might, by having lots of specialists in different places or different conditions, you might end up getting that adaptive variation anyway. So what I'm trying to say is, if you're doing a conservation plan and you're looking at only general species, you might not find this kind of result in your kind of study system. We also looked at neutral genetic variation, and here we can see that if you just use the distribution information, you actually don't get above the red line for most species. So that's not ideal, and some species you do really poorly. If you use the surrogate variables, you can see that actually improves it for most species, and then of course our genetic variables. When you directly use the data you're interested in, then of course you're going to achieve your targets. So this is really exciting, and this is really encouraging. So then my next postdoc, we explored some of this work further, and we found that actually, this whole thing doesn't generalize everywhere. So you've actually got to think about where or when these things may or may not work. So if the European Alps is easy mode, you've got enormous climatic gradients, very strong barriers to gene flow. So if you're going to be getting strong patterns of, spatially structured patterns of adaptive neutral genetic variation, it's going to be in an environment like that. The Iberian Peninsula is totally different. So this is a place that has had very complicated climatic history, where you have populations expanding and retreating during repeating cycles of glacial expansion. And what that means is just, genetically wise, everything's all over the place. It's like a mosaic. And you don't get these nice, clear kind of ideas like isolation by distance, where things that are further apart are just more different from each other. So what we looked at was 7 amphibian species, 3 reptile species, and we used microsats, if you know what that is. And as you can see, these are the distribution of the species in the data, and it's quite different. Some are really narrow ranging, other ones are much broader. And so what we have here is we generated for each species a separate prioritization where we selected sites. So we selected 1 site, 2 sites, 3 sites, 4 sites based on different rules. One of the rules was maximizing genetic diversity. And so that's what the white line shows here. So this is a curve that kind of summarizes the results across multiple different curves. So if we're able to achieve a higher value on the y-axis, we're getting more genetic diversity for a fewer number of sites. So so the white line's our best if we're using genetic data. If we're using geographic variables, so that's based on the idea that we're spreading out our prioritization spatially evenly across the study area for where the species is found. Then what we have is we have that orange line. We also looked at resistance distances. So that's kind of like a measure of so places that are separated by less suitable habitat have a higher resistance distance between them. So if you can think of like geographic variables as Euclidean distances, these are kind of like least cost path distances, sort of. And then we've got random, where we just randomly selected sites and did that like a whole bunch of times to account for stochasticity. So basically the white line is the best we could do. The blue line is if we're really dumb. And we can see that the orange line is, it's like smack bang in the middle. It's not really doing as well as we might hope. If this is really effective surrogate, we'd hope that the orange and the blue lines would be really close to the white line, but that's not the case. And what's really interesting is, in conservation, we have to have very limited budgets, which means, often we're not dealing with like situations over here on this side of the axis, we're over here, we've only got a few sites that we can look at. We can see the difference between the blue line and the orange lines. You know, they're very similar, so you might not do much better than random. So certainly, you know, that might improve things, but you know, you'd like to do better than slightly just above random, I hope. So, you know, what this means is, you know, genetic data can provide exceptional value in certain situations where these other surrogate variables don't hold up. The caveat is though that you don't know that unless you actually have the genetic data. Okay, so we've looked at lots of different surrogates and we've talked about how they compare. But, you know, if we use different surrogates, how does that affect our conservation plans? So in one of the papers I'm working on now is we're looking at different approaches for incorporating connectivity in conservation planning. So lots of different ways have been developed to account for connectivity when you're developing spatial prioritizations. And we wanted to see whether different approaches meaningfully alter your prioritization when you consider all the other stuff you normally consider when you're doing the spatial prioritization. So we've got a whole lot of data for Washington State, 261 bird species. We've got land acquisition costs, existing predicted area data. This area is a multi-use landscape. You know, there's urban areas, agricultural areas, you know, lots of different land use types. And there's also multiple different ecosystem types. So, you know, you've got forests as well as dry shrublands and that kind of stuff. So this is the costs, and the black bits are existing protected areas. Now there's a cost of 0 because they're already protected, so they're 0. So we looked at 8 different approaches, and so I'm just going to walk you through how to read this because it is a bit overwhelming. So each panel is a prioritization based on a different approach. So the light grey stuff, so we'll just look at total area for a moment. The light grey cells are places that weren't selected at all. The orange, and the dark grey cells are existing predicted areas, so they're the same. So the orange, the blue, and the yellow were places that were selected by the prioritization. So, so the dark grey was locked in, but the orange, the blue, and the yellow, those are the priority areas that prioritization selected. So we can see here the orange and the yellow, and there's like a few blue bits, but those are the places selected on the total area approach. And we can see that, so if it's orange, that means it's a place that was selected by this prioritization, but also at least one other prioritization. If it's yellow, then these were places that were selected by all prioritizations. So we can see that there's a lot of commonalities, like, you know, there's some places, there's quite a few places that were selected under all different approaches. And under the blue approach, that's only found in this approach. So if it's blue, those are pixels that are only selected when we use this approach. So critically, what we did was we generated a baseline prioritization first using suitable targets for the species, and we calculated the cost of that. And then we said, well, let's say we add an additional budget of 15 or 20%. Let's try and increase connectivity as much as possible while still meeting those targets for each of our 300 odd species. And we'll see what happens then. So, you know, this is still a valid multi-species approach that's accounting for ensuring that each species gets a suitable amount of habitat. But given that and given an extra room for improved connectivity, we're looking at how that changes our results. So what we can see here is the total area approach. So the idea behind that is really interesting. So the idea is if you have more habitat, you get larger population sizes. Larger population sizes means more dispersing individuals. More dispersing individuals means more connectivity. Therefore, if you just get lots of land and you have species that are very generalist and can, you know, can exist under a wide range of different habitat types, and you also have species that can disperse very easily across your landscape, this could be a really valid approach that, you know, just that gets you a whole lot of different places and, you know, increases population sizes. But it may not be too suitable, you know, if you've got habitat specialists, because it's not accounting for the fact that, you know, if you have different habitat types, you might be doubling up on the same habitat type, so you're not really improving the connectivity of, you know, those specialists that are found in more costly areas. So the species representation approach, what that does is that says, let's try and improve the average amount of habitat for each species, given this extra, you know, 15 or 20% budget that we've allocated just for connectivity. And what we can see is we do see some commonalities between these two, but we can see, you know, these places down here that are, you know, that's basically where the cheapest areas in the study area are. We can see there's much fewer of those, and that kind of spreads it out further as well to try and improve, you know, provide more habitat for species over here and up here. You can't see it, but this is the boundary length approach. So if you use mark sand, then you'd be very familiar with this. The idea is we want to try and minimize spatial fragmentation by creating large, big, distinct reserves. And this approach is really good if you have species that are if you have species that can't disperse easily between reserves, and you need species that need like a good buffer zone from anthropogenic impacts. We have a human pressure approach, which is the idea that species have a harder time dispersing through human modified landscapes. So if you have a greater degree of human modification, then that forms a barrier. So here we're trying to find connected areas that have low anthropogenic impact. This is a similar idea, but we've actually done this one for based, instead of just using broad scale human footprint indices, this is specifically based for the species in the study area. So knowing that maybe roads aren't as bad for other ones, that weighting differently, we can see some similarities here. Focal species is the idea you have one species that you really care about. That works well when you actually have one species you really care about. Here we use the greater sage grouse, and you can see that it kind of gives you similar results to these ones. Habitat heterogeneity is discontinuities in vegetation cause issues for connectivity, and environmental similarities, the idea that connected gradients and heat connectivity. And that can work well if you've got a lot of habitat specialists because they can't move through different environmental types. But if you have generalists, then they can go anything wherever they want. So you really want something like that instead. So I'm running out of time. So what I'm going to do is I'm just going to say, we found that each of these different approaches give you different results. There are some similarities, which is surprising. So these approaches based on very different assumptions get very similar answers. Other ones, these two are basically different ways of saying how human modification impacts connectivity. They give us broadly similar stuff. And we measure the performance of them. So I'm going to skip that because we don't have time. If we run out of questions, I can go over that then. But thank you so much for listening to me. And so basically, I just want to say, there's many ways we can improve conservation decision making. I went over 3 better algorithms, use cost-effective data, and reliable surrogates. Of course, there's many more. If you want to know what other stuff we could do, that's also a good question time. But yeah, thank you so much.